P-wave, good morning everyone. 68 question. The protein perforin is released by. So this question came from immune system. So in general, in immune system, the blood cell will play important role. That is white blood cells, also known as leukocyte. Leukocytes. In general, these leukocytes has two types, like B lymphocytes. In leukocyte, we have different types of white blood cells. In that, lymphocyte play important role in immune system. Lymphocytes, so in which lymphocytes we have two types, B lymphocytes and T lymphocytes. So this B lymphocytes which mainly helps to produce antibodies to fight against pathogen in body fluid. This antibody fight against pathogen in body fluids. So this type of fighting against pathogen by involvement of antibodies is known as humoral immunity. Now coming to T lymphocytes. T lymphocytes generally we have two types of cells, like TH cells, means T helper cells, and TC cells, cytotoxic T cells. So in that TC cells play important role. Generally this TC cell which secrete perforin, a protein, perforin. So not only TC cell, along with TC cell there is one more white blood cells are there. They are nothing but natural killer cells, NK cells. So along with cytotoxic T cells and these natural killer cells, both are secrete a protein called perforin protein. And this protein which makes pores. Pore means here holes. Pores are holes on plasma membrane of infected virus cells, infected or target cells, or tumor cells, which make pores or holes on plasma membrane of infected or targeted or tumor cells. So hence they cause the cell lysis. That is called cytolysis, means cell death. Cell death. So that's the importance of this perforin protein. So this perforin protein which only act on infected or targeted or tumor cells only, in which make the pores or holes. So by that the cell will completely die or lysis. So this role was performed by this perforin protein, not only synthesized from cytotoxic T cell but also synthesis from the NK cells, natural killer cells. So right answer here, third one. Here next one, 69 question. Here choose the mismatch pair. Here we have to identify incorrect pair. Amine hormone, example is thyroxine. Peptide hormone, oxytocin. Protein hormone, here giving testosterone. Steroid hormone is progesterone. Let's analyze this explanation, which came from chemical nature of hormones. Generally hormones are in two chemical nature, like steroidal hormone and proteinous hormone. Steroidal or proteinous hormones. Here steroidal hormone means they are lipid hormone. Here proteinous hormone means water hormone. So in general, in steroidal hormone, the hormone which include like adrenal cortex hormone and also very important gonads hormone. Gonads hormones. So adrenal cortex hormone and gonads hormones are included in steroidal hormone. So then what are the adrenal cortex hormone? So in general, adrenal cortex hormone divided, generally adrenal gland is divided into two important parts like that. So we have adrenal glands on the surface of kidney. So which have like this cap-like structure, gland is there, adrenal gland, in which in anatomically which is divided into two part, outer part and inner part. So this outer part is called adrenal cortex. This inner part is called adrenal medulla. Now this adrenal cortex part only produce steroidal hormone. In this adrenal cortex, there are three layers are present. From outer to inner, there are three layers are present. So in which the outer one is called zona glomerulosa, and second one is middle one is zona fasciculata, and the innermost layer in adrenal cortex, that is zona reticulata. So why? Because each and every layers which produce their respective hormones. For example, this zona glomerulosa layer which produce mineralocorticoid hormone. The primary mineralocorticoid hormone here, aldosterone, very important. So aldosterone is a primary hormone of mineralocorticoid which is produced from the layer of zona glomerulosa. Now coming to zona fasciculata. So this zona fasciculata which produce glucocorticoids hormone. The main glucocorticoid hormone is cortisol. Now coming to zona reticulata. So this zona reticulata which produce sex steroid hormone. Sex steroidal hormone. So the main sex steroidal hormone is androgonic here. Androgonic steroid hormone. Do you know this sex steroid hormone or androgonic steroid hormone which helps in growth of axial growth of axial hair and facial hair and pubic hair during puberty. So we know that, so once the child will enter into puberty, adolescent age, there is a growth of various hairs like axial hairs, facial hairs, and axial hair, facial hair, and pubic hairs. So this growth of hairs is due to presence of this androgenic steroidal hormone, and which is produced from the zona reticulata of adrenal cortex hormone. Remember all these very important hormones of steroidal hormones. Now coming to gonadotropin hormone. Generally gonads means the reproductive organs. So we have in male gonad is testis, here female gonad is ovary. The testis which produce important hormone, that is testosterone hormone, and here ovary produce two important hormone. They are estrogen hormone and progesterone hormone. Estrogen and progesterone hormone. So all these hormones play important role during sexual reproduction. So these two hormones like testosterone, estrogen, and progesterone, all our hormones are gonad hormones. And gonad hormones also example for which type of they have chemical nature means steroidal hormone. So if you analyze this statement here, one gonad hormone is there, so that is testosterone. But testosterone is not a here. Testosterone is a steroidal hormone, but here giving protein hormone. So this is what mismatched pair. Okay, so that hence it is a right answer. Let's here see steroidal hormone, progesterone. See, just now we discussed progesterone is a steroidal hormone, but testosterone is also steroidal hormone. But in question giving protein hormone, no, this statement is wrong. This pair is mismatched. Okay, let's we analyze some importance of protein hormone and water soluble hormone. So rest of the hormones what we have, so everything, each and every hormones are included in this proteinous hormones. Okay, clear now? Right, so we have to identify mismatch pair. So testosterone is not a protein hormone, it is a steroidal hormone. So here next one, 70th question. Consider the following statement type of questions. Here assertion and reason type of questions. Colostrum is absolutely essential for newly born baby. It contains several antibodies that protect the newborn from initial source of infection. So let's analyze this statement. So during lactation, after birth, during lactation period, and immediately after parturition, after delivery, immediately after delivery. So mother start producing initial milk. That initial milk is nothing but colostrum, and these colostrum milk are rich in very important antibodies. It's rich in antibodies. So the main important antibodies it contains, that is IgA, immunoglobulin A antibodies, very important. It's colostrum milk which contain IgA antibodies, and these IgA antibodies which provide passive immunity, very important, passive immunity, which provide passive immunity to protect infant, to protect newborn baby or infant from various vulnerable and digestive tract diseases, against various pathogen or especially against environmental pathogen. The newborn baby or infant get protected by having this antibody, IgA antibody, which is rich in this colostrum milk in initial day, mother producing after delivery in initial day. Okay, so this is, and both statement colostrum is absolutely essential for the newborn. Yes, why? Reason is why? Because it contains several antibodies. So which antibody is IgA antibodies, and what is the importance? Which provide passive immunity to the newborn baby for initial source of infections. So both assertion and reason type of state both are correct statements. So hence the right answer is 1. Okay. So next 71. Here match the following type of questions. So in list 1 giving various contraceptive devices and their importance. Okay. Let's discuss some analysis of about these questions here. So these questions which is came from the health. In reproductive health, it is completely framed from contraceptive devices. Contraceptive device. So, do you know what is contraceptive device? The device which is prevent pregnancy. Such devices are called contraceptive. so which prevent pregnancy or avoid pregnancy. So we use some devices. Such devices are called contraceptive devices. In general, we have various types of contraceptive devices, but here we have to analyze only these four devices. So here coitus interrupts. Here coitus interrupts means, here we will discuss about first coitus interrupts. So here coitus interrupts means, so it is a traditional natural method of contraception. Natural method. It is a traditional natural method of contraception. So during sexual intercourse, we have to remove our penis before we ejaculate the sperm into the female body. So we have to interrupt the coitus, and it is a natural method. No use any kind of devices. Okay. So but here in this condition, we don't want to release our sperm into the female body. Before releasing sperm into female body, we have to interrupt this coitus, sexual intercourse. So this type is called coitus interrupt. So here it is a natural method. So hence first is fourth one. Now coming to diaphragm. So here this diaphragm is a physical barrier method used by female. So diaphragm is nothing but it is a physical barrier, physically, which prevent the entry of sperm into the female body. Physical barrier. So generally this diaphragm are used by female only. So hence this is also known as female condom. Okay. So this female condom are nothing but diaphragm which mainly used to prevent the entry of sperm into the female body. So hence B is first one. Here B is first. Now coming to progesterone. So very important, progesterone. So it is a hormone releasing IUD, very important. So here hormone releasing IUD, intrauterine device, progesterone. So hence here second one. So generally which hormone is released here? IUD means intra device. So this intrauterine device we use, that is progesterone. So which release hormone, which releasing hormone, which hormone means progesterone hormone. Okay. And coming to Saheli. So generally Saheli is a contraceptive oral pill. Saheli is a contraceptive oral pill. So about Saheli, this Saheli drug was manufactured or prepared by CDRI, Central Drug Research Institute in Lucknow, and it is a weekly pills. So weekly once we have to take, and remember it is a steroidal pill, steroidal drugs. So CD Saheli is a weekly once we have to take this Saheli to prevent contraceptions, and this was manufactured by the CDRI, Central Drug Research Institute in Lucknow, and it is a steroidal hormone, very important. So it is a contraceptive oral pill. So by this statement we have to analyze, and then here right answer is here 4125, 41 here 4125. So right answer is third one. Okay, next 72 question. Trisomy of 23rd chromosome in male. Here asking about in male. So what result in? So these questions which is from Mendelian disorder. In Mendelian disorder, this will comes in genetic disorder. So we'll analyze genetic disorder. So all this Turner syndrome, Down syndrome, Edwards syndrome, Klinefelter syndrome, all this comes under genetic disorder or Mendelian disorders. So let's analyze. So do you know in human, in male or female, 23rd chromosome in male, that is XY, is a sex chromosome. So do you know, remember, so we have 23 pair of chromosome. Among 23 pair of chromosome, 22 pair of chromosomes are autosomes. Remember, autosome. From 1 to 22 pair, always autosome chromosome. But 23rd 23rd pair of chromosomes is allosome. Here allosome means sex chromosomes. Only 23rd pair is allosome or sex chromosome. And this sex chromosome is different in male and in female. In male, the sex chromosome or 23rd pair is XY. In female, XX. So like that we have. Now here, in context for this question here, this disorder which come from only 23rd pair of chromosome. So here 23rd pair of chromosome nothing but sex chromosomes. So hence in male 23rd chromosome is XY. In female 23rd chromosome is XX. Remember, in female. Okay, right. Let's analyze. So here one word is given, trisomy, and here what is trisomy? So trisomy means here extra chromosome is added. That condition is called trisomy. And one more important term is there, that is monosomy. So monosomy means one chromosome is deleted. deletion of one chromosome is called monosomy. So here we have formula 2n minus n, but in trisomy we have 2n plus 2. So that is the differences between monosomy and trisomy. In trisomy, sorry, one here. In trisomy, one chromosome is added, but in monosomy one chromosome is deleted. Okay, but in question here asking only trisomy, so we will analyze. It means that in generally we have 2n is equal to 46 chromosome in human being. If one chromosome is added means we have 47 condition, so that condition is called trisomy. For example, from 2n is equal to 46, one chromosome is deleted means minus 1, then we have 2n is equal to only 45. So this condition is called monosomy, this condition is called trisomy. This one monosomy and this one trisomy. So it means that trisomy has one extra chromosome, that is 47. In monosomy it has one less chromosome, that is 45. We will discuss all these analyze. Okay then, so here what exactly trisomy of 23rd chromosome in men. We are asking about trisomy in men. We will analyze both here. So yeah, so here I am writing in male. Here in male 23rd pair of chromosome is X here. In female 23rd chromosome is XX. Fine, right. So here what happened, in which trisomy occur in male, if there is a trisomy. So in trisomy means 2n plus 1. So here means trisomy means having one extra chromosome, just now we discuss one extra chromosome. So it result to formation of, generally we have 44 autosomes, and here already we have one pair of sex chromosome, that is XY, but one more X is added here. So here the condition, the karyotype here 44A plus double XY, this condition will be found. Okay, so it means that there are 47 chromosomes are present. This condition is called Klinefelter syndrome, and these Klinefelter syndromes occur only in male, very important, in only male. The condition here in which 44 autosome plus double XY, so one extra chromosome is added here, hence it is a trisomy. Now coming to female here, so we analyze in female likewise. So in generally in female we have monosomy. So the condition which is occurred, that is monosomy. Do you know monosomy 2n minus 1? So it means that lose one chromosome, one X. Why? Because female always have both XX chromosome. So here in which one X chromosome is loose. So when one chromosome is loose from the sex chromosome, then 44 autosome as it is here X and 0, very important. So here 0 indicate no X chromosomes. Only one, we have only one sex chromosome, that is X chromosome. So what is the condition here? So what is the karyotype here? 44 autosome plus X0. So there are only 45 chromosomes are present. But in Klinefelter there are 47 chromosomes. But here 45 chromosome only present. And this condition, this syndrome is called Turner syndrome. Remember, so Turner syndrome occur in female due to deletion of one X chromosome in monosome. So this Turner syndrome in female, the karyotype is 44A plus X0. So total number of chromosome is 45. But in male trisomy, here karyotype is 44A plus XXY, and that condition is called Klinefelter syndrome, which is occur in male. So here in asking trisomy of 23rd pair, here trisomy of 23rd pair chromosome. So hence the right answer is Klinefelter syndrome. So what about Turner syndrome? This one in female. Just now we discussed now in female. So what is the condition here? 44 autosome plus X0. So total number of chromosome is 40, occur in female only. Remember, very important. And now coming to, we will analyze these two, Down syndromes and Edwards syndrome. So what exactly this Downs and Edwards syndrome we will analyze here. Yeah. So here the Down syndrome and Edwards syndromes are both are trisomy condition, but this trisomy which occur in autosome chromosome, very important. So hence it is a autosomal disorder. But here Klinefelter and Turner syndromes are allosomal disorder. Remember, but Down syndromes and Edwards syndromes are trisomy condition, but it is a autosomal disorder. Why? Because and this extra addition of chromosomes occur in only autosome chromosome. So we have 1 to 22 pair of autosome chromosome now. So there only we have seen these conditions. For example, in Down syndrome, so where Down syndrome it has means 21st chromosome. So trisomy of 21st chromosome. Remember, trisomy of 21st chromosome, it leads to cause Down syndrome. It means that generally at 21st we have only pair AA chromosome. But here what happened? One extra A chromosome is added like this. One extra A chromosome is added. So now here at 21st chromosome we have seen trisomy condition. Now this condition is leads to cause Down syndrome. And coming to Edwards syndrome. So Edwards syndrome also trisomy, but in which chromosome means very important 18th chromosome. It means that generally in 18th chromosome we have like this AA. In pair only we have chromosome, but here what happened? One extra chromosome is added here like this, A chromosome. So here also trisomy, but this trisomy condition which occur at 18th chromosome. Now this condition is called Edward syndrome. And one more syndrome we have actually which is not mentioned in this question here. So that is Mahadyuth syndromes. So in Mahadyuth syndromes, it is also autosomal trisomal disorder. So it is occur at 13th chromosome, 13th pair of chromosome, 13th autosomal chromosomes, 13th autosomal chromosome. It means that generally 13th autosomal chromosome has only pair like this. But here what happened? One extra A is added like this. So here we have three set of chromosome at 13th chromosome we observe. Now, so this is also trisomy occur at 13th autosomal chromosome, and this type of condition is called Mahadyuth syndromes. Mahadyuth syndrome. So like Edward syndrome, Down syndromes, and Mahadyuth syndrome. All are the other syndromes is due to autosomal trisomal disorders. So remember. And Klinefelter syndromes and Turner syndromes. Both are allosomal or sex chromosomal disorder. In that, Klinefelter syndrome is trisomal disorder, and Turner syndrome is a monosomal disorder. So here in 23rd, 72 question asking about trisomy of 23rd chromosome. Hence, the right answer is only here Klinefelter syndrome. So we analyze all this Mendelian disorder in detail. Okay. So here next one. 73rd question. If the blood group of parents are homozygous for A and homozygous for B blood group, here we analyze here. Parents blood group here giving parents blood group. So one parent blood group is homozygous A blood group, and one more parent blood group is homozygous B blood group. Okay, fine. And these blood groups are not expected here. This blood group is not expected in their children. So which blood group is not expected in their children? If the parent having homozygous A blood group and homozygous B blood group. Here, let's analyze this MCQ. Homozygous A blood group means IA, and homozygous B blood group means IB. Okay, so both are homozygous. So in homozygous condition, we have only one gamete. So hence here IA, from which we can get IB. So if we cross these two, we can get IA, IB. So children has which blood group? IA, IB means AB blood group. Their children has AB blood group. Okay, fine. AB blood group. And here in questions asking, so these blood groups are not expected in their children. So which blood group is not expected in their children means we have four blood group now. We have four blood group like A blood group, B blood group, AB blood group, and O blood group. But their children has which blood group here? AB blood group. So which blood group is not expected to their children means we have to remove this AB. Then rest of the blood groups are not expected in their children like A blood group, B blood group, and O blood group. So here right answer is AB O blood group. Why? Because their children has AB blood group. Okay, remember. Okay, so let's analyze this 74 question here. Study the following and choose the correct statement. Here we have to choose correct statement. So there are four statements are given. Cumulative effect of two or more genes and a single phenotypic trait is known as polygenic inheritance. Karyotype of Turner syndrome. In previous class only we discussed about Turner syndrome. Here giving karyotype of Turner syndrome is XXY. The longest gene code for the muscle protein, that is dystrophin. Highest number of genes are located on 21st chromosome. Let's start analyzing of these questions. So here we have to analyze these questions in detail. Yeah. So here first know that about Mendel. So according to Mendel, what he said, and Mendel said now, so that completely deviate from some rules and regulation we will discuss here. First, according to Mendel, one gene express only one character. One gene express only one character, and one gene always have only two alternatives. These two alternatives are called alleles. And every character also has two alternatives. Every character also have two alternatives. Those alternatives are called contrasting trait. Very important. So according to Mendel, Mendel said one gene always express only one character. One gene always have only two alternatives. They are called alleles. One character always have two alternatives. They are called contrasting trait. For example, here we analyze. If you take one gene, that is T gene. So T gene express which character? Stem height character. And T gene has only two alternatives, like capital T and small t. And stem height has only two alternatives. They are called traits. Means they are tall trait or dwarf trait, like that. So here tall and dwarf represent what? Contrasting trait. Capital T, small t represent what? Alleles. Here only single t is represent gene. Stem height represent character. So like that Mendel said every gene express only one character. Every gene has two alternative forms. They are called alleles. And every character has two alternatives. They are called contrasting. This is as per according to Mendel. But this concept was deviated or excluded. How means? Here see. So here I am writing deviation of Mendel's. Yeah, deviation of Mendel. Deviation of Mendel's. So here how they are deviated means one gene has more than two alternatives. But according to Mendel, one gene has only two alternatives. But here one gene has more than two alternatives. Such alleles are called multiple allele. Multiple allelism. So this multiple allelism concept was deviated from the Mendel. For example, here there is a gene which is present on the ninth chromosome of human being. The gene is called I gene, and this gene is only responsible for blood group. And this gene, every gene has two alternative as per Mendel, but here deviated. How means this I gene has three alternatives, means three alleles it has. If more than two alleles, such type of allele is called multiple allele. Hence what are those three alleles? Like IA, IB and I. Like that there are three alleles. So completely this multiple allelism is deviated from the Mendel according to as per Mendel. Okay, so this is the best example we have seen this multiple allelism in ABO blood group. Okay, fine. One more deviation here we will discuss. Yeah. So one more deviation is every gene, one gene express only one character. That is the according as per the Mendel. But here one gene which express more than two character. One gene express more than two character. Such type of gene is called polygenic genes. Polygenes, sorry, not sorry, pleiotropic. We use the term. Pleiotropic means in which one gene express more than two characters. Here we have few example of pleiotropic genes like phenylketonuria is a disorder. This phenylketonuria disorder is due to one gene express more than two characters. Phenylketonuria. It is a pleiotropic gene. In pleiotropic gene means here remember. So one gene which express more than two characters. Okay. Now coming to one more deviation here we have. Yeah. So here yes. Now third deviation here I am writing. So here generally we know that one gene express one character as well as one character must and should be express only one gene. But here more than here there are multiple genes which express only single characters. Here generally as per Mendel one gene express only one character, but here we have more genes, multiple genes which express only one character. See, just now we discussed in pleiotropic one gene express more than two characters, that is pleiotropic. But here one character is expressed by more than one gene, so that is polygenic. So they are quite opposite to each other. Pleiotropic and polygenic both are opposite to each other. How means here one gene is express only one character as per Mendel, but here one gene express more than one character, that is pleiotropic. So one character is expressed by more than one gene, that is polygenic. So this statement is called polygenic. So in name only it is suggesting poly means more, gen, polygenic genes. So more genes express only single character. So best example we have, that is human skin color. See, human skin color. Why? Because so not all human beings have same skin color. So we have different skin color. So that different skin color is due to this polygenic inheritance. So not only that, even though human height, so no individuals having same height we have different heights of human beings. So this human height is also example for polygenic inheritance. So this is all about the important concepts related to these statements. Okay, so remember, according to Mendel, what he said and deviation of Mendel. So we have, there are three deviation of Mendel. One is multiple allelism, second one is pleiotropic gene, and third one is polygenic inheritance. Remember. Okay. Yes. Now let's start analyze all these four statement. Okay. Here, first statement: cumulative effect of two or more genes means poly. So this polygene express only single phenotypic trait. Here, phenotypic trait means single character. So if more genes express single characters, such genes are called polygenic inheritance. So this statement is right. Just now we discuss. Now coming to second statement: karyotype of Turner syndrome. In previous MCQ we discussed about Turner syndrome. Here Turner syndrome karyotype is AA XX1. No, this statement is wrong. Why? Because in Turner syndrome, what is the karyotype here? 44 autosome plus X0. Only 45 chromosomes CP have. So this statement is wrong. So whose karyotype this one? 44 autosome plus XXY. Who's this? So this is the Klinefelter syndrome karyotype. Klinefelter syndrome. So Klinefelter syndrome karyotype is 44 autosome plus XX, and in which we have 47 chromosome, which is generally seen in only in male, and that Turner syndromes are seen in female only. Female. Okay, analyze. And this statement is wrong. Second statement. Now coming to here, longest genes code for the muscle protein, that is dystrophin. Yes. So we will analyze these two statements, and this two statement analyze from the Human Genome Project. Very it is a mega genome project, and this mega genome project which started from 1991 to 2003. Actually 2003 is a end period, but due to here some reasons, which is extended to 2006. Okay, so in mega genome project here, so here we will discuss about Human Genome Project in brief. This Human Genome Project, finally it revealed that there are in human genome contain approximately 30,000 genes. Remember, in Human Genome Project, which revealed that there are 30,000 genes are present in human being. How much? 30,000 genes. Okay, now? Right. So here, yes, highest number of genes. Here I am writing highest number of genes is located on chromosome 1. Very important. In Human Genome Project, they reveal that. So which chromosome has highest number of genes? Highest number of genes located on chromosome 1. So how many genes are located on chromosome 1 means, here this number is very important: 20968. So 2968 genes are located on chromosome number 1. So here chromosome number 1 has highest number of genes. Now which chromosome has lowest gene? Here lowest number of genes are present on Y chromosome, male chromosome. Y chromosome. Okay, so Y chromosome has lowest number of genes. How much genes it has means, only 231 genes are present on Y chromosome, and 2968 genes are present on chromosome number 1. Hence it is the highest number of chromosome containing highest number of genes containing chromosome is chromosome number 1. Remember, very important. And do you know the largest known human gene? Here I am writing just, yeah. So here the largest known human gene is nothing but dystro, and this dystrophin gene is located on X chromosome. Remember, so on X chromosome only this dystrophin gene is located, and this dystrophin gene is the largest gene in human genome. Okay, but remember, the highest number of genes present on chromosome number 1, lowest number of genes present on chromosome Y, but the largest genes present in chromosome X. Don't confuse. Here X chromosome contain largest gene, that is dystrophin. Chromosome number 1 contain highest number of genes. How much means? 2968. Lowest number of genes containing chromosome is Y chromosome. Remember all these things. Okay, so how much generally this dystrophin gene has 2.4 millions of base pair. 2.4 millions of base pair. Here base pair means two nucleotides together form one base pair. No? So like that there are 2400 millions of base pair. So for example, this dystrophin genes contain 2.4 millions of base pair. Remember, very important this one. So all these are the concept related to the human genome project. So let's analyze third statement. The longest gene here. The longest genes code for the muscle protein, that is dystrophin. Yes, this statement is right. Dystrophin is a known largest gene which is present on which chromosome? X chromosome. So this third statement is right statement. Now coming to fourth statement, highest number of genes are located on 21st chromosome here giving. No, not 21st chromosome. On which chromosome the highest number of gene is presented? First chromosome. Which chromosome? First chromosome. So 21st is right, wrong answer. Only the highest number of genes are located on only chromosome number 1. How much means 2968. So this statement is also wrong. So here there are three statements are wrong. Second, third and fourth. So we have to choose correct statement, that is first one. Yeah, sorry. Here third statement is also right now. The longest gene code for the muscle protein dystrophin. Yes. So first and third one. Yeah, first and third one. Okay. So this is all about the human genome project and their analysis, and according to Mendel and deviation of Mendel we will discuss. So next class we will continue from the 75th questions. Thank you. N, Q, R, S and T wave. P, Q, R, S, T. So we are discussing about T wave now. So here T wave, abnormally tall T wave, which indicate very important hyper. So remember, abnormal tall T wave. See, for example here, generally T wave must and should be dome shape. Then it become normal. If it is abnormally increased like this, abnormally tall T wave, if it is formed, then it leads to cause hyperkalemia. So here hyperkalemia is nothing but more or excess potassium level in blood. It indicate what? Excess potassium level in blood. So if excess potassium level in blood, that condition is called hyperkalemia. So hence the right answer is second one. Okay, so next one, 79th question here. Geological name of European honeybee. So here we have different types of honeybees. Generally, do you know here honeybee show polymorphism? Very important. Polymorphism. It means that the individuals of the same species, individual of the same species existing in three different structures. If any species individual of this same species exhibit in three different structure, such type of species are known to be as a polymorphism. So best example is honeybee. Why means this honeybee exhibit in three different forms, in which queen honeybee, and queen is a female, it is a sterile, sorry, it is a fertile female honeybee. And one more honeybee, that is drone, it is also fertile but it is male honeybee. And one more female honeybee but it is sterile, that is workers. So worker is a sterile female. So like that, a single species has three different structures, hence this honeybee shows poly. And we know that the queen is the only important and a honeybee in which in queen generally it has a diploid polyploidy, and here it has haploid, and sterile female also like worker also having diploid. Remember this queen which developed by fertilization of egg, but very important, this drone which developed by unfertilized egg, very important here, the drone which developed by unfertilized egg. So if any organism which developed by without fertilization, such organisms are called as a parthenogenesis. So here among these three honeybee species, honeybee types, which species become parthenogenesis means that is drone, male. And here coming to sterile female, that is worker, and it is formed by fertilized only, and which is developed by fertilization of egg. And both females are developed from fertilization of egg, but male developed from unfertilized egg. Such organisms are known to be as a partheno. Okay, and here generally this queen lay egg. The primary role of this queen is lay eggs only. Do you know per day there are 2000 eggs are lay per day. And coming to function of main role of drone, which is mainly helps, which is play important role only for mating with queen, mating purpose. After mating this drone will be die. And worker play major role among this honeybee, and which collect nectars and which prepare honey. Like this various works will be performed by this worker honeybee. Okay, like that we have different types of different single species has different forms, hence we have like three types of honeybee organisms. So but here in worldwide we have different types of honeybees. So for example here giving some, we have to analyze this explanation. Yeah, So here giving 4 important species. So Apis indica. Here Apis indica generally it is a Indian honeybee. And here Apis dorsata. So Apis dorsata is a rock bee, also called the rock bee honey, which is generally present on rock. And Apis mellifera, it is a European honeybee. European honey, or also we can use Western honey, or it is a Italian honeybee. Also we can use Italian honey. So hence the European honey or Western honey or Italian honey is Apis mellifera. Okay. So next and the last question from zoology, that is 80th question here. Fish byproduct used in cleaning the wine. So which fish byproduct we use for the cleaning the wine or beer, we will discuss. So we know that these questions from fisheries, and fisheries is nothing but it is one of the important topic in strategies for enhancement of food production. So coming to in fisheries, generally fishes produce various byproducts. And these byproducts are useful for various purpose. In that, most important fish byproduct is here isinglass. So this isinglass byproduct which is used as a clarifying agent. Here clarifying agent nothing but which clean the wine and beer, which helps to clean the wine and beer. And one more important fish byproduct, so that is shark green. Here second one, shark green, or generally this shark which is obtained from shark fish skin, and which is used as a sand for polishing wood and ivory. Here this one. And one more important, that is fish guano. See here, this fish guano used as highly nutritious food. Fish guano is a highly nutritious protein food for poultry. It's a highly protein rich food for poultry only. So like that we have various fish byproduct, but here in this context the fish byproduct used in cleaning the wine is nothing but isin, and it is used as a clarifying agent to clean the wine and beer. So right answer is third one. So by this we have completed zoology paper analysis with of EAPCET, which is held in 5th May 2026. Thank you. Have a nice day. Consider the following statement type of questions. One species develop into two or more species. It's called divergent evolution. Statement 2. Analogous organs suggest divergent evolution. Okay, we will analyze this statement in detail. So here about evolution. From evolution only this question will come in evolution. Evidence of evolution. So there are various evidence was proposed by various scientists. In here we will discuss divergent evolution. So here divergent evolution, which mainly refers the differences between closely related population, and these differences between closely related population leads to species. So often this speciation due to adaptation to different environment. So here in speciation what happened, in which one species, here from one species develop into one species develop into two or more than two species, leads to divergent evolution. So hence the statement 1 is correct. And remember this divergent evolution which also leads to homologous organ. Here in homologous organ, the organ origin is same, but the organ perform function is different. Report. Origin of organ is same, but their function is different. Let's discuss some example for homologous. Here best example is mammalian forelegs. So for example here, like whales and bats and cheetah and human. So here we have various mammals, and the origin of forelimb is same. Here in which all mammals origin of organs. Which organ here? Forelimb organ is same, but they perform different function. How means? For example here, whales perform this forelimb for swimming, and bats perform this forelimb for flying, and cheetah perform for running, and human being use this forelimb for locomotion or movement. So like that, origin of organ is same, but they perform different function. This type of organ is called homologous organ. It is basic is divergent evolution. And one more example also we have here for divergent evolution. So that is thorn of bougainvillea and tendrils of cucurbita. In these two organisms, the origin of this organ, here organ like thorn and tendrils is same. Means from where they origin? From the axillary bud of stem. the origin from auxiliary body of state, but they perform different functions. How means thorn perform defense function, they perform defense mechanism against dosing herbivores. Now here tendrils perform function like it gives structural support for climbing. See here tendril perform climbing purpose and thorn perform defense functions. So like that different function but their origin is same. So like that we have different functions and same origin. Such type of organ is called homologous organ. Now coming to third and most important example for divergent evolution, so that is Darwin finches, so which is observed in Galapagos Island. And also one more example for divergent evolution, that is Australian marsupials. So these are the various example for divergent evolution. So let's start some analysis of this two statement here. See, so read once. Statement 1, if one species develop into two or more species, it is called divergent evolution. Yes, sure. Means it leads to speciation. For leading speciation is the main reason is due to this divergent evolution. So this statement is right. Here analogous organ suggests divergent evolution. No. Which organ suggests divergent evolution? Homologous organ suggests divergent evolution. But then analogous organ suggests what? Analogous organ suggests convergent evolution. So in convergent evolution, the origin of organ is different, but the organ function is same in analogous organ. Remember. So statement 2 is wrong. Hence, right answer is third one, in which statement 1 is correct, but statement 2 is incorrect. So next one, 76th question. The view of inheritance of acquired character of Lamarck was opposed. So these questions also came from evolution, in evolution, theory of evolution. So there are various theory of evolution was proposed by various scientists. So in that first we will discuss the theory of evolution was first attempted by Lamarck. So about Lamarck. So Lamarck was a French naturalist and he proposed a theory. The theory is inheritance of acquired character. So he proposed a theory called inheritance of acquired character. And he proposed this theory based on a principle. Based on one principle, he proposed this theory. That principle is the use and disuse of organ. Main principle on which he proposed this theory, that is use and disuse of organ. And do you know, Lamarck wrote one important book, that is Philosophy Zoologica. His book Philosophy Zoologica. So in 1809 he wrote this book. Okay. And what actually this theory, that is inheritance of acquired character, explain. And this theory which mainly explain here I am writing. This theory mainly which explain here the characters which is acquired here. The character which is acquired during its lifetime, those characters are, so here the character which is acquired during its lives in response to environment, and those characters are passed down to next generation, he said. So if any organism which acquired character during its lifetime, and those characters must and should be passed to the next generation according to this theory. And for that he took two examples. And here for example he took giraffe by using this use and disuse of organ. So he took example for giraffe for use organ. How means, here we will discuss how means. So generally giraffe stretch its neck to get its food from tall tree, and this stretched neck here, stretched neck, it acquired a character during its life cycle, and these characters are passed on to the next giraffe, next generation. Why? Because this neck is continuously used. Hence this example given by used organ. And I also explain disused organ in snake. Generally snake has four limbs, but due to generally these snakes doesn't use four limbs for their locomotion. So what happened, day by day these four limbs completely reduce four limbs. Hence due to reduce four limbs they shows burrowing nature. So here these four limbs are disused, it is not used in its life form. Hence these four limbs completely reduce, and these characters also comes to the next generation by disuse organ. Why? Because here disuse organ is four limb. Okay. So on this principle he proposed. And finally this theory like inheritance of acquired character which is proposed by Lamarck was disproved or opposed or rejected by a scientist called August Weismann. We will discuss about August Weismann, how he disproved. Okay. So about August Weismann, he disproved or opposed Lamarck theory of inheritance of acquired character. How we will discuss. Generally, August Weismann, he was a German scientist, and he disproved by his famous experiment. Conduct one famous experiments. So that is he cutting tail of mice for 22 generation. Remember, for 22 generation. But in every generation there is a tail generation will come in their next generation. Tail means there is no tailless mouse will come, even though we can cut the tail. So finally, what he want to conclude that, and by this experiment he proposed a theory. August Weismann proposed a theory. So the theory is continuity of germplasm theory. Very important. The theory of continuity of germplasm theory. So this theory with this continuity of germplasm theory explained here. This theory explained in germ cell. Germ cell here nothing but reproductive cells. The changes in reproductive cells here. He said that changes in germ cells or reproductive cells only inheritable, but not somatic cell changes, but not inheritable of somatic cell. So it means that if any changes will occur in somatic cell which does not inheritance to the next generation, if any changes will occur in germ cells only, they are inheritable. Here tail is nothing but it is a somatic changes. If you cut the tail means it is a completely tail, it is a somatic organ. If we cut means that is not inheritable. But he said that by this theory he want to say that so if any changes will occur in germ cells and they are only inheritable to their next generation. Otherwise no somatic cell will inheritable to their next generation. So this theory was proved. Okay. So this is all about the August Weismann. So finally August Weismann only opposed the Lamarck of acquired inheritance of characters. Okay. Right. So here right answer is Weismann. So here next one 77 question. Cynognathus is an intermediate form between. So these questions from like connecting link questions. So here Cynognathus is a connect. It is we know that it is a well-known extinct mammals like reptiles. It is extinct mammals like reptiles. So hence the Cynognathus is a connecting link between reptiles and mammals. So here we need to remember some other connecting link between various organisms. Here note. So but remember Cynognathus is a connecting link between reptiles and mammals. Hence first one is right answer. So like do you know virus? So viruses is a connecting link between non-living organism to living organism. So the connecting organism between non-living and living that is virus. And one more important euglena. So euglena also a connecting link between plant and animal. Remember very important between plant and animal. The intermediate form between plants and animal that is euglena. Hence it is act as a connecting link between plants and animals. So likewise protospangia, protyrospangia is a intermediate form of between protozoa and porifera phylum. So between this protozoa porifera, the intermediate form of organism that is proteospangia. And one more connecting link need to remember that is Neophilina. So this Neophilina is a connecting link between annelids and mollusca. Annelid phylum and mollusca phylum. And very important here, peripatus. So peripatus is a connecting link between Annelida and Arthropoda. So the connecting link organism between Arthropoda, Annelida and Arthropoda that is peripatus. And one more important connecting link are the intermediate form of organism that is Belano. So this Belanoglasses is the intermediate form of organism between non and cords. And one more last that is lungfish. So lungfish is a connecting link between fishes and amphibians. So like that we have various intermediate form between organisms. So but in contest here Cynognathus is the intermediate form or it is a connecting link between reptiles and mammals. So hence the right answer is first one. Along with you also remember all these connecting link are intermediate form of organism between organisms. So here next one 78 question here ECG tall T wave indicate what? So we will analyze ECG instrument here about ECG. Here ECG means electrocardiograph. It is an instrument to measure electrical impulses of heart. And this ECG instrument was discovered by a scientist called Antho. Very important. ECG instrument was discovered by a scientist called Antho. So here in this ECG generally form three waves. In ECG there are three waves of formants like this. See the dome shape here, and then here below down and above, again down and again above, like this, and again dome shape. So like this ECG electrical impulse will form in these waves. So here we will identify. So here it is a P wave, Q, R, S and T wave. So here we have three different types of waves. So that is P wave, and second one is C. This triangle completely Q, R, S waves. And then again we have T waves, QRS waves and P waves. So like that we have three different types of waves. So here we need to remember each waves indicate what and what are their importance we will discuss. Okay. Yeah. See here P wave. About first we'll discuss about P wave. Sorry. Yeah. So generally what is the shape of the P wave? Dome shape. So these P waves indicate what? This P wave indicates atrial systole. So we know that during heartbeat there are three important major steps will occur, that is atrial systole, ventricular systole and depolarization, like atrioventricular relaxations. So in that first atrial systole will occur just 0.1 second. This atrial systole also called atrial contraction, or also called atrial depolarization, atrial contraction and atrial systole. Generally here see this is a four chamber of the heart, right side and left side, right side atrial and right side ventricle, left side atrium and left side ventricle. So when there is a blood in this two atrium chamber and then it undergoes systole, means contractions, and this contraction of both atrium occur only just 0.1 seconds. 0.1 second only this atrial systole will occur. So our atrial contraction or atrial depolarization will occur. This P wave indicate. And if there is a abnormality in P waves, so what are the abnormality means? Just abnormality leads to some disorder. By observing this P wave impulses, we can identify some disorders, like for example here the P wave, enlarged P wave. For example here this P wave is there now. For example it is like this, enlarged P waves, and these enlarged P waves which indicate enlargement of atrium, which indicate what? Enlargement of atrium. If there is a enlargement of atria, if we can identify as enlarge of P waves. If P waves become inverted P waves, and this indicate defect in SA node, which indicate the defects in SA node, sinoatrial node. If, for example, we know that here we have in right atrium, we have in right atrium at the right side corner of the right atrium, here we have SA node, sinoatrial node we have. So if there is any defect, so we know that it is act as a phase makers. Why? Because from here only the impulses will generate. If any defect in SA node, it was indicated in this inverted P wave. If for example inverted P wave means if like this, if P waves will form like this inverted, then it indicate there is a defect in this SA node. Okay, so like that there is a indication of disorder of P waves. So remember, P waves indicate generally atrial systole, atrial contraction or atrial depolarization. So it occur only 0.1 second. So likewise we will discuss what are the indicators of QRS wave here. Yes. So and then triangle shape QRS wave. QRS waves. So this QRS waves generally in which shape? Tri in shape. So in which very important, this QRS wave indicate what? Which indicates here. Now in P wave indicate what? This both atrial systole now, and this indicate ventricular systole, are also called ventricular contraction, or also we can use another term, that is ventricular D. So which occur generally 0.3 seconds. And if any disorder or if any disorder which is related to QRS and which indicate, like for example here, if enlarged R waves, if there is a enlarged R waves, for example here we have now a top R waves, if still it is more enlarged, then this enlarged R waves which indicate enlarged ventricles. So just now we discussed if enlarged P waves which indicate enlarged atrium. So hence here if there is enlarged R waves, then it indicate enlarged ventricles. And very important here, if there is a enlarged Q wave, if there is an enlarged Q wave which indicate very important, that is myoinfraction. Here simply myocardial infraction is nothing but heart attack. For example here Q there no Q waves here like this. But if it is enlarged like this, if it is enlarged Q waves like this, then it indicate what? Myocardial infarction, means heart attack. So very important. If there is a enlarged R waves, it indicate enlarged ventricle. But if there is a enlarged Q wave, so enlarged Q waves which indicate myocardial infarction, that is heart attack. So in which what happens generally, blood flow is completely blocked. So in which there is no flow of blood to the heart, hence that leads to heart attack. It is due to, and this heart attack condition can be observed by seeing this impulse, that is enlarged Q waves. Okay, right. Next, yes, next third one, T waves. So here T waves also dome shaped structure, dome shape. Here see, T waves also dome shaped like this, and this T wave indicates what? This T waves which indicate ventricular dash. Just now we discussed QRS waves indicate ventricular systole, but this T wave indicate ventricular diastole, means here in which ventricular get relaxed, ventricular relaxation. Also we can use another term, biology term, that is ventricular re, we can use these terms, and generally which is occur 0.4 seconds, and ventricular systole occur 0.3, and 0.1 for atrial systole. Like that 0.8 second is needed for each heartbeat. Okay, and say here T waves, if any defect in T waves which leads to various disorders, like for example here, if flatted T waves, if here generally T wave like we have like this na T wave, but if it is flatted T waves, means like this, flatted, like flatted T wave become flatted, then it indicates what? It indicates insufficient oxygen to the heart. It indicates if there is a flatted T waves impulse will generate, then it indicates insufficient oxygen to the heart. So we can analyze if flatted T waves are there, there is a insufficient oxygen are reached to the heart muscles. If inverted T waves, inverted T generally we have like this dome shape. If it is inverted like this, if we in T waves like this, here I'm showing you, if there is T waves like this, inverted T waves, and these inverted T waves which indicate very important, that is myoischemia. So myocardial ischemia is due to, which is we can analyze by seeing inverted T waves, myocardial ischemia. So here myocardial ischemia means blood flow is reduced, but in myocardial infarction complete blockage of blood flow, so it leads to heart attack. Okay, but in inverted T waves it is a sign, it is a signal for in future it may be heart attack. So in which what happened, blood flow is reduced, not completely blocked, but in which blood flow is reduced, so heart muscle doesn't receive sufficient amount of oxygen to perform its function. So that condition is lead to cause myocardial ischemia is. So we can analyze this problem by seeing inverted T waves. Just now we discussed in here Q waves, inverted Q waves, if sorry enlarged Q waves which indicate what? Myocardial infarction, heart attack. But here inverted T waves which indicate myocardial ischemia. And not only that, one more important, if there is a abnormally tall T waves, for example like generally we have T waves like this, just wait, generally we have T waves like this dome shape. If it is abnormally tall like this, T waves only, if it is abnormally tall like this abnormally tall T waves. So okay, so finally here P wave and Q R S and T wave, P Q R S T. So we are discussing about T wave now. So here T wave, abnormally tall T wave which indicate very important hyper. So remember abnormal tall T wave. See for example here generally T wave must and should be dome shape, then it become normal. If it is abnormally increased like this, abnormally tall T wave, if it is formed, then it leads to cause hyperkalemia. So here hyperkalemia is nothing but more or excess potassium level in blood. It indicate what? Excess potassium level in blood. So if excess potassium level in blood, that condition is called hyperkalemia. So hence the right answer is second one. Okay. So next one, 79th question here. Zoological name of European honeybee. So here we have different types of honeybees. Generally, do you know here honeybee show polymorphism? Very important. Polymorphism. It means that the individuals of the same species. individual of the same species existing in three different structures. If any species individual of this same species exhibit in three different structure, such type of species are known to be as a polymorphism. So best example is honeybee. Why means this honeybee exhibit in three different forms, in which queen honeybee, and queen is a female, it is a sterile, sorry, it is a fertile female honeybee. And one more honeybee, that is drone, it is also fertile but it is male honeybee. And one more female honeybee but it is sterile, that is workers. So worker is a sterile female. So like that, a single species has three different structures. Hence this honeybee shows poly. And we know that the queen is the only important and a honeybee in which in queen generally it has a diploid polyploidy, and here it has haploid, and sterile female also like worker also having diploid. Remember this queen which developed by fertilization of egg, but very important, this drone which developed by unfertilized egg, very important here, the drone which developed by unfertilized egg. So if any organism which developed by without fertilization, such organisms are called as a parthenogenesis. So here among these three honeybee species, honeybee types, which species become parthenogenesis means that is drone, male. And here coming to sterile female, that is worker, and it is formed by fertilized only, and which is developed by fertilization of egg. And both females are developed from fertilization of egg, but male developed from unfertilized egg. Such organisms are known to be as a partheno. Okay. And here generally this queen lay egg. The primary role of this queen is lay eggs only. Do you know per day there are 2000 eggs are lay per day. And coming to function of main role of drone, which is mainly helps, which is play important role only for mating with queen, mating purpose. After mating this drone will be die. And worker play major role among this honeybee, and which collect nectars and which prepare honey. Like this various works will be performed by this worker honeybee. Okay, like that we have different types of different single species has different forms. Hence we have like three types of honeybee organisms. So but here in worldwide we have different types of honeybees. So for example here giving some, we have to analyze this explanation. Yeah, so here giving four important species. So apis indica, here apis indica generally it is a Indian honeybee. And here apis dorsata, so apis dorsata is a rock bee, also called the rock bee honey, which is generally present on rock. And apis mellifera, it is a European honeybee, European honey, or also we can use western honey, or it is a Italian honeybee, also we can use Italian honey. So hence the European honey or western honey or Italian honey is apis mellifera. Okay, so next and the last question from zoology, that is 80th question here. Fish byproduct used in cleaning the wine. So which fish byproduct we use for the cleaning the wine or beer, we will discuss. So we know that these questions from fisheries, and fisheries is nothing but it is one of the important topic in strategies for enhancement of food production. So coming to in fisheries, generally fishes produce various byproducts and these byproducts are useful for various purpose. In that most important fish byproduct is here isinglass. So this isinglass byproduct which is used as a clarifying agent. Here clarifying agent nothing but which clean the wine and beer, which helps to clean the wine and beer. And one more important fish byproduct, so that is shark green. Here second one, shark green, or generally this shark which is obtained from shark fish skin, and which is used as a sand for polishing wood and ivory. Here this one. And one more important, that is fish guano. See here, this fish guano used as highly nutritious food. Fish guano is a highly nutritious protein food for poultry. It's a highly protein rich food for poultry only. So like that we have various fish byproduct, but here in this context the fish byproduct used in cleaning the wine is nothing but icing, and it is used as a clarifying agent to clean the wine and beer. So right answer is third one. So by this we have completed zoology paper analysis with of EAPCET which is held in 5th May 2026. Thank you. Have a nice day.